Hello, my name is Pora Kim. I'm happy to present my recent study, the FusionNeoAntigen in Vaccine Congress 2024. So the title of today's talk is Systematic Bioinformatics Studies for the Development of the Targeted Therapeutics for Human Fusion Genes. So I will introduce about my fusion gene studies, and in the second part, I will introduce about FusionNeoAntigen database. So again, my name is Pora Kim. I'm working in the School of Biomedical Informatics at the University of Texas Health Science Center at Houston. So let me introduce my overall study so far. So I have interest in developing therapeutic target in human genome with diverse biological mechanisms or events. So far I published many different kinds of topic related to personalized medicine oriented therapeutic target knowledge base. So the major research topic for me is the fusion gene, and I also have interest in alternative splicing events, and also I published diverse of RNA editing events or cancer gene or cancer features or other disease information. So here, using diverse mechanism or diverse algorithm or approaches or pipelines of bioinformatics in the biomedical informatics, we are proposing to systematic human genome-based therapeutic target in different ways. So I hope this resource can be some knowledge base or resource or reference for each mechanism study. So let me introduce about fusion gene. So as many of you know, in human cancer, there are many different types of mutation, but based on the size, we classify those. So the shortest one is point mutation. We call it single nucleotide variants. And sometimes we see diverse lengths of indels, and we often see the copy number variation. And the largest size is, we call it as structural variants. But we can say that the fusion gene is when the breakage happened inside of the gene body, not other intergenic region or other region, then we call it as the fusion gene. So we can say fusion gene is a kind of byproduct of the structural variants. So when there happen like multiple DNA double strand breakage happened simultaneously, and then our cell tried to fix it, but it failed, and then it will keep the wrong rearrangements like this structural variants. And these structural variants have the breakpoints inside of a gene body. So it made a fusion gene. And then following the central dogma, it can make a fusion transcript and it can form fusion protein. And then we can target these fusion proteins using drug screening or other ways. But today I can introduce about neoantigen. So first, let me introduce about our resource usage. So Fusion GDB is the first and unique functional annotation database of human fusion genes. Yeah, this number, I think it should be updated. Anyway, so yeah, as you can see here, almost of the words are using our fusion gene database for their study, and we pioneered diverse ways of fusion gene analysis and studies in many diverse studies, like as shown in here. So Fusion GDB is second version updated by using AI model. Anyway, so we provide the very comprehensive knowledge of the fusion gene. So like what is the known breakpoint so far, and which breakpoint is the most frequently happened fusion breakpoints. And then we developed the fusion AI model to distinguish fusion gene or not. So using that, we provide some genomic area feature And also we provide our own methodology to identify the coding potential to form fusion protein. And we also developed the fusion FGPure, unique visualization of the functional feature when the fusion gene happen. And we also provide the fusion gene transcript sequence and fusion protein sequence. So UniProt is importing our fusion protein sequence. And we also provide annotation about fusion PPI events, the impact and the related drug disease. So yeah, this is overall stat. And then last year we published the fusion protein database. So using AlphaFold 2, we predicted the structure of about 3.5 fusion proteins. And then we made our own criteria to select some confident structure. And then for 68 chosen proteins, we performed virtual screening. We provided some potential small molecules. So this is FGPure. So after studying many years for the fusion gene, I learned that there are this kind of representative action mechanism of action in the cell of the fusion genes. So I thought if we can have some unique visualization to infer the fusion gene function, it would be helpful. So I developed like this. So this is showing like a fusion gene, two gene and DNA level, mRNA level, protein level. So these are the 39 protein functional features provided from UniProt. So when we move this red line to breakpoints, we can infer what kind of functional features will be retained or are deleted. And the Fusion AI is a CNN model to distinguish a fusion gene or not. So it reads the plus minus 5 kb from the breakpoints over two gene. So it finds, it suggests the possibility. So when we compared with the other NGS-based tools of prediction of two fusion genes, somehow we think our AI model can efficiently remove the false positive. So I recommend you, when you predict fusion gene, then run other multiple tools of prediction from NGS, and then one more time running our tool. So yeah, from invitation of the STAR protocol, we published this Fusion AI protocol. And this is, so based on when we studied the Fusion AI model, somehow we thought if we can utilize this high feature importance score region, which means that sequence area has a key feature distinguishing fusion gene or not, we may find some genomic evidence of the microhomology mediated end joining. So we try to find the first top high feature importance region, and we try to check whether they're between two genes, whether they have some sequence similarity, sequence homology. Then we found this number of microhomology events. So here and then across the all chromosome, we see that the seventh position have some high C sequence. And then we compared with the number of FusionNeoAntigen. As you see here, this chromosome, these yellow chromosomes highlighted here are having more number of observed FusionNeoAntigens compared to expected number. And the Fusion network we published this year. So Fusion network, we thought still the kinase is important because it is the key target in cancer. So most fusion kinases are usually driver mutation in patients. So we thought it need to arrange or assess the impact in the clinical aspect of the individual kinase in pan cancer fusion gene network. So among 500 kinases, almost of them are involved in fusion genes. So first, we made a network out of, first we made this kind of noble numerical values for each gene involving in 100,000 fusion genes, like number of cancer types, number of partners, number of breakpoints, this kind of metrics. And then we try to perform the dimension reduction using PCA or variation autoencoder. And using the ranking based on this dimension reduction, we studied the network propagation as using those high rank genes as the seed gene for the network propagation. And then from there, we calculate the impact of individual kinase to form the pan-cancer fusion genes. And somehow we made this kind of ranking. The outer one is our score, fusion network score. And the second layer is the number of sample involved in fusion gene. So this is for the kinase belong to tyrosine kinase group. The other group, you can visit our website. And these days, we are also trying to understand the aspect of the protein breakage tendency, not in the DNA level, but protein level. So this kind of problem is hard to get the data, but we are trying and understanding the relationship with the evolution conservation. So I hope we can wrap this work quickly. And these days, we are establishing, trying to establish a novel bioinformatics pipeline to target fusion proteins. We named the fusion target. So the pipeline looks like this. So it just the starting point is the patient's fusion breakpoints in the DNA level. Then we annotate ORF frame, and then we make a full-length transcript, and then we predict the protein sequence and the predict structure, and then we perform our specific virtual screening with some special way, and then we apply other criteria. And then we worked for the EWSI1, FRI1, which is the driver mutation in Ewing sarcoma. And somehow we suggested some molecule, then it looks working. And this summer we are working for two database, which is kinase fusion DB. And this is a novel knowledge base of human kinase fusion gene. And there are 7,000 kinase fusion genes. And then after checking, we have 1,700 in-frame fusion gene. It makes 1,400 fusion proteins. So for these, we provide a diverse, very unique annotation and virtual screening results also. So we are arranging all potential ranking of the kinase inhibitors. And recently we studied we wanted to get the landscape of therapeutics of the fusion gene of proteins from PubMed search. So we searched PubMed with this number like 1.5 billion times and also 1.8 quintillion times of abstract search based on number of fusion gene, number of synonyms, gene synonym, in head gene, tail gene, and 14,000 drugs. So we found for this one, fusion proteins, we found 10,000 abstracts, and we are waiting the result of 100,000 human genes. And for these, we're providing diverse annotation. For this one, I think it will be useful, and we can also use for our study. From now on, I will present about FusionNeoAntigen. So this is a resource of fusion gene-specific neoantigen. So I will introduce this work based on the bioinformatics aspect. So neoantigen is can be identified by T cell if it presented in the tumor cell, outside of tumor cell. So anyway, so this is neoantigen is presented by tumor cell, not normal cell, because it's mutations. So whatever the mutation or RNA editing or diverse fusion gene or whatever other some abnormal sequence, if it can be translated or not translated. Anyway, so it can make a certain degraded short peptide. So we name it neoantigen. So, and then our T cell will recognize it and it will attack. So we want to, if we use this one and develop some cancer vaccine, then we think we can somehow help the boost of the T cell recognition and the immune efficiency. So the overall process of the neoantigen prediction is like this. So first, we do the biopsy for patients, and then we do the NGS sequencing data, and using PIME4MX algorithm, we identify multiple aberrations, and then we check. Here we call the variants, and then we check also expression. And so for diverse indel or SMV or fusion, alternative splicing or these things, we have to analyze the open reading frame and then infer the peptide. And then using chemical approach, we have to generate the peptides. And then we can check the immunogenicity from using some algorithm or in the cell level or mouse or real patient level. So fusion neoantigen is studied from our resource of fusion GDB second version. It includes about 102,000 human fusion genes. So it has a lot of breakpoints. So considering all multiple breakpoints in each gene and multiple gene isoforms, we check all in frame, the open reading frames. And then we found 16,000 in frame fusion genes. So these can make about 82,000 full-length mRNA sequence. And then these can make 43,000 proteins. So out of these, we made 13,000 unique fusion breakpoint amino acid sequence, like plus minus 13 amino acid. And then we run the usual tool to predict neoantigen, net MHC pen, or deep HLA pen. And then we found this number of about 500,000 fusion neoantigen interacting with HLA-1 and 20,000 neoantigen interacting with HLA-2. So this is showing the example of BCR-ABL1. The middle is a breakpoint. So, and to provide more reliability, we also adapted the AI 3D structure prediction approach. So here we use a RuTafold and not AlphaFold 2, because when you come, AlphaFold 2 requires certain lengths of protein, but RuTafold, it can work for the short length. So we choose this one. And then using site map of Schrödinger package, we predicted the active sites in individual predicted neoantigen structure. And then so we checked the interaction between HLA, known HLA proteins and the structure and our predicted neoantigen. And we also somehow we checked out of 14 fusion protein sequences, we found about 4,000 fusion genes are interacting, making fusion gene with the cell surface protein partner. So from here, these proteins, and then these proteins are from 544 fusion genes. They are retained transmembrane domain. So for these, we predicted their structure. Somehow we are thinking this can be some target of a CAR-T therapy. So this is showing the pipeline of fusion neoantigen prediction. So it's very similar to general way. And this is, so far, there are not many studies done for the fusion gene and neoantigen. And this one shows the general statistics of fusion gene, how many fusion gene, how many translated, how many HLA, this kind of stat. And this one shows the number of samples that have potentially expressed the fusion neoantigen from our study. So we show the top cases here. And then this is the 3D complex between neoantigen and HLA molecule of these famous fusion genes. So if you visit our website, you can see diverse information. So first, you can get the fusion protein breakpoint sequence plus minus 13 from the breakpoints. And this is the multiple sequence alignment of the identified neoantigen at the same breakpoints. And this is fusion protein sequence. And this is the structure of cell surface fusion protein. And this is the result of a prediction of fusion neoantigen by running MHC pen and the deep HLA pen. And this is a structure of short peptide of fusion breakpoint area. And this is a binding score between HLA molecule and the neoantigen. And this is the result of domain retention investigation of the cell surface fusion protein. And this table shows a subcellular localization prediction using other tool. Yeah, so future direction is, these days we have an interest in pediatric sarcoma patients because it's very aggressive patient. And sometimes I have some question from patients like, anyway, I think it's very aggressive tumor. So I want to help the kids. So I hope using high immune power of kids, I hope we can use this fusion neoantigen vaccine efficiently to treat pediatric cancer. So thank you for listening.